at this point, it becomes very important that you have taken in all the lessons that we've been discussing up until this point, the concept of ions existing in different concentrations on one side of the channel or the other, and the concept of ions being ready to flow in one direction or another, and especially the idea that sodium will readily flow into the cell or that potassium will readily flow out of the cell, and exactly why that happens, exactly why it happens that way. So we talked about the resting membrane potential. We gave some different ideas about exactly why it happens the way it happens. But it's important to note that membrane potentials can actually be changed. They can be changed by, number one, anything that alters an ion's concentration on one or the other side of the membrane. And number two, anything that changes an ion's permeability, or its ability to flow from one side to the other. Let's talk about the first case first. Anything that alters an ion concentration on one or the other side of the membrane would be something that changes, for example, the number of potassium ions or sodium ions inside of the cell, or the number of potassium or sodium ions outside of the cell, or perhaps even the number of proteins inside of the cell. Anything like that would start to change the membrane potential, perhaps in subtle ways, perhaps not by much. Perhaps instead of -70, it might be -68 or -65, but that would be a significant enough change that the cell would no longer function the same way that it used to function. So one example of the way that this could occur would be if, for example, remember that outside of the cell there's a very small number of potassium ions. There are certain conditions in which all of a sudden the potassium ions would actually be increased outside of the cell. This would be a disorder, and this would be a disaster. The body would no longer, if the body was no longer regulating its potassium ions outside of the cell, then the membrane potential would change significantly. So potassium ions, sodium ions, chloride ions have to be regulated at a particular number in order to make everything work correctly. So that's kind of a rare instance. It would probably be a disorder. There are certain injuries and so on that would lead to this problem. But number two, the second situation actually happens all the time. Something that changes an ion's permeability would be something as simple as a channel. And one of the channels we're going to be looking at, and one of the first sort of a case we're going to consider, is the idea of a chemically gated channel, also called a ligand-gated channel, or more generally would be called a stimulus-gated channel. So this chemically gated channel will open when a certain chemical binds. So what you'll see here is there's a closed ion channel, and there will be some kind of a ligand or some kind of a neurotransmitter that will bind to this receptor outside of the cell. And that will allow for the channel to open, and at that point the ion will flow through. An example of a channel like this would be something like a acetylcholine-gated sodium channel. So you've named the ligand in the first part, which causes the opening of the channel, and in the second part you've named the ion that flows through. So if you had, for example, a ligand-gated potassium channel, then when that particular ligand binds to those receptors, it would cause the potassium to flow out. Another version of this would be a mechanically gated sodium channel. In that case, basically a pressure change in the cell membrane would cause the channel to yawn open and sodium would flow into the cell. So you get the idea here. There's a nomenclature that's involved here where you have the naming of the channel takes on two parts. The first part talks about what gates the channel. The second part talks about the ion that flows through. Very common mistake that students make is that they will start to call channels, they will start to call them, for example, sodium-gated channels. That's incorrect. Sodium itself is not gating the channel. In this case, it's some kind of a ligand. It's a mechanical stimulus. It's a neurotransmitter that's gating it, that's causing the gate to open. The ion is the thing that flows through. So it's sodium, chloride, calcium, potassium, whatever the ion is the second thing that is named because it's the thing that flows through. So be very careful that you know exactly what's gating the channel and then what flows through secondarily. Okay. All right. This is just another example of a stimulus-gated channel. In this case, it is a channel that is permeable both to sodium and potassium. When the neurotransmitter binds and the channel opens, sodium flows in and potassium flows out. And just so you know, the drive is stronger for sodium. There's a few different reasons for that, but in a general sense, sodium tends to enter faster than potassium flows out. The second type of channel we're going to be looking at is a voltage-gated channel. There's two, for our purposes, there's only two voltage-gated channels that we actually consider, a voltage-gated sodium channel or voltage-gated potassium channel. Again, note the nomenclature. The gating is mentioned first and the ion is mentioned second. So a voltage-gated sodium channel is shown here. And what happens with a voltage-gated channel, although it's not an easy concept to get, I understand that it's not easy to understand this, so you have to kind of sit with this information for a while, is that a voltage-gated channel will open when there is a change in voltage. So with a voltage-gated channel, it says it right in the name, voltage gating. A change in voltage actually causes the channel to open. So if you notice here then, this is the way that a cell is typically set up, where you have positive charges lined up along the outside of the cell, negative charges lined up along the inside of the cell, and then something changes. Something changes so that there's a reversal. All of a sudden there are negative charges outside, positive charges inside. That's actually going to cause this channel to change its configuration and flip open. Again, it did not require a neurotransmitter. It required a change in the membrane potential itself. And since this particular channel is permeable to sodium, what happens in this case is that the voltage gate opens and sodium flows in. There are also examples of voltage-gated potassium channels. And just so you know, there is a third type of channel, which is called a leakage channel, which is open all the time. The only one that we consider in the nervous system is a potassium leakage channel. It's actually a very important channel that allows potassium to leak out at small increments on a regular basis that actually keeps potassium at a slightly higher concentration outside of the cell than would be otherwise. Down the line, way down the line, when you talk about the cardiovascular system, you're going to take into consideration some sodium leakage channels, which have a very, very important role in the pacemaker potential of the cell. So there are three types of channels then. The ligand-gated or chemically gated or stimulus-gated channels, the voltage-gated channels, and then the leakage channels.